Yeah, today I'm going to present an Cytoscape app for the prediction of the disease gene and disease association, and I also provide some functions for event collection. And first of all, I would like to show the methods for ranking the disease candidate. And as you know, the biologists usually face with a problem of getting many candidate genes from susceptible chromosome region, for example. So the goal is how to rank the a large set of the candidate gene. So there are so many methods have been proposed for this problem. And here today I present an app based on network-based ranking. And so there are so many network-based algorithms for this problem, and as shown in the literature, the RWR algorithm is the best one compared to another, and it is the best compared to the algorithm based on the homogeneous network of gene. However, when we integrate the gene network with a kind of the disease severity network, so the RWR will modify to the RWRS, and it gets a better performance. And here's briefly information about the RWR algorithm. Basically, it is a kind of algorithm that propagates the disease information over the network. And finally, each gene in the network are given a score which represents a degree of association between the candidate gene and disease of interest. And in 2012, I published the GPEC. It's based on the RWR algorithm. And this year, I published the HGPEC is based on the RWRH algorithm for the same problem. And here's the main difference between the HGPEC and JPEG. Basically, they based on the JPEG based on the homogeneous network of gene, and the HGPEC based on the heterogeneous network of gene and disease. So there are some little difference between the HGPEC and JPEG. They are all can predict the novel disease gene association, but HGPEC can also predict the disease association, and it can work with disease without molecule basics. And they all have some function like visualization, event collection, and also the annotation. And here's the HGPEC design. It looks more complicated, right? However, the input is a kind of the heterogeneous network of gene and disease, and user can provide the candidate gene in various ways, like enable of the training gene or the chromosome region, or freely provide any candidate sets. And then after ranking, the candidate gene can be ranked, and we can visualize the gene and disease based on the network, based on their score assigned during the ranking. And the highly ranked gene and disease can be provided with the annotation as well as the evidence from the PubMed or OMIM or any other data that I show here. And here's some function of JPEG. The first one is visualization. We can visualize any association between gene or between disease or between disease gene association based on ranking and based on the network topo. And here's the function of the event collection and annotation. The novel association can be supported with the annotation from ontology, from the cake, and also protein complex. And the novel association can be supported with the event collection from the PubMed or OMIM or something else, some other database. Okay, that's all about the HGPEC. Thank you for that attention.